Hello, I'm presenting our study on testing alternative approaches in constructing biological datasets for DNA-based diatom bioassessment. Metabarcoding studies on diatoms often face the biases arising from, first, the incomplete reference libraries resulting in complete taxa lists, and two, clustering DNA sequences into operational taxonomic units based on an arbitrary similarity value and which are unique to the given analysis, thus hardly transferable among studies. So we hypothesized that by not using taxonomic assignment, clustering, or denoising of the individual sequence units, relevant biases can be removed and a more standardized bioassessment can be promoted. In order to test our hypothesis, we produced 4 types of biological dataset. 1 taxa list with microscopical analysis, and 3 datasets with metabarcoding. Among these, we created 1 operational taxonomic unit list with the model pipeline using a 95% similarity threshold. We produced an exact sequence variant list using the DADA2 pipeline and the thorough denoising algorithm. And finally, we produced individual sequence unit list with only basic quality filtering and the replication steps. The biological lists were then used to develop an index based on the ecological profiles of the given taxonomic units along a reference environmental gradient. Index values were then calculated on a test dataset, and results were correlated with the reference gradient. Based on several observed parameters, we found that the ISU index performed the best and the OTU base performed the worst. When analyzing in details, we found that there were some abundant OTUs in the OTU dataset containing ecologically different sequences. The difference of these sequences regarding their ecological optima could be revealed by the ISU index. We can conclude that the taxonomy and clustering-free approaches may favor a standardized and comparable DNA-based bioassessment due to the following reasons. First, analyses in ISUs are reproducible and transferable between studies. Second, ISUs are consistent among studies as their identifier is the DNA sequence itself, unlike in the case of OTUs. And third, ISUs allow a higher resolution on intraspecific or on intrapopulation level. It is, however, important to note that ISUs are not ideal to study, for example, rare biospheres where sequencing errors may strongly bias the results. In that case, DADA2-like denoising algorithms producing exact sequence variants or amplicon sequence variants have great importance. So finally, I would like to thank all my colleagues and institutions participating in this study, and thank you for your attention.